Welcome, first semester medical laboratory students. I am absolutely thrilled to have you here. Today, we're skipping the slow introductions and we're just going to drop you straight onto the clinical lab bench. As a technologist, you know blood is that specialized circulating tissue you'll be handling the most. It's literally your ultimate window into a patient's health.
Okay, let's dive into this. When you pick up an anticoagulated whole blood sample tube, what are you actually looking at? Because it's definitely not just some red liquid. I want you to mentally visualize this right now. You are holding a highly complex, living tissue. It's composed of a liquid extracellular matrix and literally billions of microscopic formed elements. And they're all interacting in perfect harmony to maintain homeostatic balance, transport respiratory gases, and provide immunological defense.
Now, to truly understand this tissue, we've got to pull it apart, right? So if we take that whole blood, collected with an anticoagulant to keep it from clotting, of course, and we pop it into a centrifuge at high speeds, we're physically separating the blood into distinct fractions. Basically, by manipulating the force of gravity, all those heavier cellular components get forced right to the bottom, while the lighter liquid components naturally rise to the top. And this brilliantly illustrates exactly what you'll be seeing on the bench. Your tube is now divided into three very distinct macroscopic layers. That top layer, that's a straw-colored liquid we call plasma, and it makes up roughly 55% of the total blood volume. Right beneath that, you'll spot a really thin, kind of grayish-white middle layer known as the Buffy coat. That's where your white blood cells and platelets hang out. And finally, resting heavily right at the bottom, are the packed red blood cells, accounting for that remaining 45% of the blood's volume. Now, what's really fascinating here is the precise chemical makeup of that top 55% layer. Plasma is, well, it's essentially an aqueous solution. It's about 93% water, but it is packed with electrolytes, transport proteins, and, this is absolutely crucial, active coagulation factors. As future lab technologists, you have got to remember this distinction. Because the blood in this specific tube was treated with an anticoagulant, the plasma actually retains its clotting factors, like fibrinogen. If you were to let the blood clot first, that resulting fluid is called serum, and all those clotting factors, yeah, they'd be completely used up.
Okay, let's move to the microscopic level and see how this builds our understanding of that remaining 45%, the formed elements. First up, we've got the erythrocytes, or red blood cells. Picture these as 7.5 micrometer biconcave discs, just packed to the brim with hemoglobin, designed specifically to deliver oxygen to tissues and carry carbon dioxide away. Next, you have the leukocytes, your white blood cells, which include your granulocytes, lymphocytes, and monocytes. These guys are your patient's primary immunological defense and essentially phagocytic sweepers. And finally, the thrombocytes, or platelets. Fun fact, these are actually tiny, non-nucleated fragments shed right from megakaryocytes in the bone marrow, and they are absolutely vital for primary hemostasis and initiating blood clots.
So, the crucial point is how all these distinct physiological components map directly to the routine hematology investigations you'll be running every single day. When a doctor orders a complete blood count, or a CBC, you are directly evaluating those formed elements, the red cells, the white cells, and the platelets. An erythrocyte's sedimentation rate, or ESR, that specifically measures how fast those red blood cells settle through the plasma under gravity. And when you run coagulation studies, like a PT or APTT, you're specifically assessing those active clotting factors we talked about, suspended right there within that straw-colored plasma layer.
Ultimately, everything you see in that tube tells a story about a living human being's physiology. It tells us about their oxygen capacity, their immune response, and their bleeding risk. You aren't just spinning tubes in a lab. You are literally unlocking critical diagnostic data. Because behind every single result is a real patient, relying entirely on your accuracy. So first semester technologists, you've got the fundamental blueprint now. Are you ready to analyze?